Hello. Today I will be discussing the genetic diversity of secondary forest. First, we need to define what old growth and second growth forest are. Primary forest, also known as old growth forest, are long-standing, undisturbed, continuous patches of forest that have a diverse number of species and alleles. Secondary forests are growing on previously cleared or disturbed land, usually through timber or being cleared for agricultural pastures, and can be regenerated through natural means such as seeds blowing on an abandoned pasture or by humans actively trying to restore a forest. So, why is this relevant? Excessive amounts of logging, especially in South and Central America, have created a situation where some countries have more secondary forests than primary forests. We can see this in the map where some countries like Brazil, Mexico, Cuba, Colombia, and Venezuela have huge amounts of forests that are less than 20 years old. Thus, understanding the forest in the modern world will help understanding second growth forest. In addition, climate change will place extreme stresses on these forests with higher temperatures and extreme droughts in a relatively short amount of time. So, these forests will need a large amount of genetic diversity to quickly adapt to these changes before they're driven to extinction. As Dr. Mendelsohn put it the other day in class, natural selection has saved more species than humans had. So we need to help natural selection by giving them a large amount of additive genetic variants so they can quickly adapt and evolve and help preserve these species in the wild. In Davies et al. 2015, the researchers looked at the diversity of V. ferruginea of old and second growth forests at two sites. The table shows the number of trees, the allele coerciousness, and expected heterozygosity for the categories of two tree species in Costa Rica. They found that the adults of the old growth forest, second growth forest, and the second forest progeny have no statistically significant difference in the allelic richness and expected heterozygosity at both sites. However, the old growth progeny were the most diverse in both species, but this is still considered a successful recuperation of most of the genetic diversity, which gives us hope in the recovery of the diversity of second growth forests. In addition, a different research team looked at the diversity at Eterupe edulis in protected old growth forest, agroforest, and secondary forest. Agroforests are forests that have regrown from human care, and in this study, both agroforest and second growth forests had fruit that was harvested. Among the 6 sites, there was little significant difference in inbreeding, allelic richness, and expected heterozygosity. Thus, this further confirms the idea that trees of secondary forests are often resilient and regaining their genetic diversity. And in the graph, we can see the 3 sites in each of the 2 columns were the 2 tree species, and there was relatively few significant differences among all the metrics measured among all the species. However, not all forests can regain this genetic variance. In this study, researchers looked at the diversity of Iterarte deltoidea in Costa Rica. The study had a main site that was broken into 3 sections, which can be seen in the picture on the right in the vertical column. You have the old growth at the bottom with all the orange, the near second growth forest, which is the section in the middle, then the far second growth forest, which is the section at the top. They also looked at a smaller site, but however that site had not enough trees to be too relevant. In figure 2, this displays on the x-axis is all the parent trees in the old growth forest, and on the y-axis is the percent reproductive contribution of these parent trees to the secondary forest. And so you can see that 50% of the reproductive contribution of the secondary forest was due to just 2 trees, which just shows that this second growth forest is not very diverse because most of the genes just come from 2 trees, which is not very good. In addition, we can look at more charts from the study, and so in the bottom left we can see a graph where the x-axis is how far into each section the trees are, and the y-axis is Tatimoto's D, which measures the diversity of a set of genes. And so you can see that second growth trees are significantly less diverse than old growth trees. In addition, we can look at the graph in the middle with the 3 charts. The axes are the same, but this time it measures saplings. So we can see that second growth near saplings are significantly less diverse than second growth far saplings, which are almost as diverse as old growth saplings. So the authors attributed this to toucans. So toucans have nested in the old growth forest, and they're making regular trips to the second growth near forest. And so this could explain why some trees are so much more popular if toucans live in these trees. And Also, there's a regular supply of gene pools from a narrow set of trees. However, in the far forest, migrating toucans or toucans with no territory could be flying in from far places into the area not controlled by the toucans of the old growth forest and creating saplings there. And so they contain pollen and seeds from a wide diversity of trees from a large geographic area. So that could be why those forest trees are so much more diverse. And this just highlights how important that pollination, seed dispersal, is in maintaining and gaining genetic diversity. This brings us to look at some of the ways that forests can be resilient with genetic variance. Having reliable pollination through animals in the wind from a diversity of trees is a key component to getting diverse genes. In addition, being near an old growth forest, full diversity tends to act as a gene pool to help give the second growth forest a wide diversity of genes. However, this doesn't always work as seen with the last study. However, some failures that can contribute to poor genetic variation could be poor seed or pollen dispersal. So if your seeds only go a few meters, or pollen doesn't spread too far, or if it's dependent on pollinators which have gone extinct, or the population has gone extinct due to deforestation, that can be a huge issue. Or forest fragmentation, we have large continuous forests being split up into small sections that are discontinuous, makes it really hard to spread these genes through these gaps in the forest. So, in conclusion, secondary forests are a major part of modern ecosystems. Genetic diversity is important to maintain the genetic variation needed to allow these forests to adapt during climate change caused stresses. We know that forests can recover this diversity and tend to be very resilient. However, this is not always the case, and it depends a lot on access to genes from diverse forests and trees. Some interesting areas to explore would be the interaction of returning pollinators and how that affects tree diversity. Like, do we need a lot of pollinators or a few pollinators? Just how exactly that pans out. And how important the rare alleles are that are lost in secondary forests. So these forests don't always get all of the alleles back, and are the few rare alleles that are lost, are those critical for adapting to climate change, or are they not that important? So those would just be interesting areas to expand on this research.